this is exactly what the microbes are doing to our patients. And the microbes have figured out a way to interact with our ability to kill them. And so I'm going to introduce you to the concepts of viable but non-culturable microorganisms, as well as introducing you to a new concept called phoenix cells. So let me get control back. So let's consider a very simple situation, a sterile pyorrhea, where you effectively have microbes in our bladder that are causing these persistent urinary tract white blood cells. But when you send that urine sample down to the lab, even though there are white blood cells present, you're scratching your head because it's negative. Well, what we now know is that there are many limitations to culture. My previous colleague has gone through many of those, and to try to recover some of our time, I'm going to skip these bullet points that reinforce that. But the end result is negative cultures have been associated with about a 4.5-fold increase risk of reinfection. As we all know from our study of outcomes, anytime a patient is reinfected, it's bad news for your statistics because it means that something untoward happened during your management of that initial case. But in point of fact, what we are now appreciating, thanks to next generation sequencing, is many of these infections are actually polymicrobial. And in point of fact, many of the microbes can resist the initial empiric antimicrobials that we prescribe for our patients to get rid of that sometimes infection that we were able to culture, but at the same time, other microbes can come about. So let's enter persister cells, or viable but non-culturable. This is principally the work of Dr. Kim Lewis at Northeastern University. He's done a lot of work trying to understand the genetics basis of persisters and what turns them on and what turns them off. But the bottom line is that at any moment of time, a microbial population, whether it be a mixed environment of many different types of microbes, or whether it be a pure culture, these 0.3% of that population are actually persister cells. So if you have 1 billion organisms per mil, about 3 million of them are in the persistent state. What this means to you, the practicing clinician, is that these cells are refractory to antimicrobials. And effectively, the microbe just sits there, folds its arms, and says, impress me, and I'll wait until the antibiotics are diluted out by the kidney and liver. And so consequently, the community that remained can revert and grow. And grow they do. They reestablish the infection. And this is often the instance when the patient comes back, again, complaining of the symptoms associated with a hallmark infection. So Dr. Lewis and his colleagues who have been studying this problem have been trying to decipher the genetics. Now enter Rita Colwell. Now, this concept is not new to microbiology. And in fact, Rita and her team actually defined VBNC back in 1982 while she was working on Vibrio. And what Rita's team appreciated, and Dr. Colwell, as many of you know, is the former head of the National Science Foundation under Bill Clinton, she appreciated that any environmental stressor, whether it be a strong oxidant, whether it be an antibiotic, whether it be simply a decrease in the soluble oxygen levels, will actually send the microbial population into a VBNC state. And in fact, the individual you see in the lower right-hand corner of the slide, Dr. Bill Kimmel at the University of Southampton, has actually taken it one step further, principally in the food micro arena, in which he showed that many of us eat that prepackaged lettuce. We're probably going to have some of it for lunch today. And what we know is that prepackaged lettuce is actually washed with about 50 part per million sodium hypochlorite, or very dilute bleach. And the consequence of that action is it sends the population of microbes affiliated with that lettuce and romaine and spinach into a VBNC state. Yet when we consume that, viable but non-culturable Listeria, Salmonella, E. coli effectively can come out. And if you happen to have a weak constitution, whether you be immune compromised or somehow other stressed, those microbes can establish an infection in your gut, and the consequence is you appear ill. This, of course, is very costly to the food industry. There have been mega recalls of millions of pounds of fresh produce, cantaloupes, all sorts of things. And Bill has now taken this one step further and has entered your arena, albeit in urinary catheter lines. And what Bill has begun to characterize is biofilm development in trying to understand whether the action of developing a biofilm on a foreign object, like the plastics that are used for catheters, whether they be silicone or whether they be some of these antimicrobial silicone catheters that are entering the market, Bill has shown or explained is why these antimicrobial urinary catheter materials have actually failed. And what he has found, and this was a paper that was published in February in MBio, is that any medical device that enters the body provides a high-risk interface for bacterial attachment and biofilm formation. That's not the news. What is the news is the fact that these microbes that are developing the biofilm, whether they be on virgin silicon or whether they be on these silver impregnated silicon matrices, actually enter into a viable and non-culturable state. And what they have shown is that why the antimicrobial materials demonstrated high utility and value in laboratory studies, it was a consequence of poorly designed lab trials that considered the impact of VBNC populations that then could predict why they failed to work in a real clinical trial. And so here what we have presented for us in this figure is effectively three different matrices that the catheter is made out of. We have silicon, we have hydrogel latex, and we have silver alloy hydrogels. And the antimicrobial nature of the matrices increases as we move left to right. And what you see from the slide is that the fraction of VBNC members within this population is actually quite high. In point of fact, within 2 hours of implant, 92% of the population in the silicon, hydrogel latex, and in the silver alloy, where the silver is actually serving as an antimicrobial perturbant, it actually sends 97% of that population into a VBNC state. And as the silver alloy will effectively elute as a function of diffusion, or it effectively oxidizes over secondary to the attack by neutrophils and other things. And what we learn is that that VBNC microbial community can come back and then wreak havoc for the patient. So VBNC and foreign materials are a real thing. And in fact, what Bill is arguing is that anytime we put something foreign in, we really need to think about what's going on with the microbial population that be resident in the environment. Now, consider what you all do. When you put in a new appliance, oftentimes you dump in a gram or two of vancomycin powder. This effectively is a strong perturbant to the microbial population, and the consequence of that act is it will send the community into a VBNC state, and the vanc is actually diluted out by the kidneys and other metabolic processes. So now we enter into the discussion of the rise of the phoenix cell. Now, this manuscript comes to us from an orthopedic group at The Ohio State University. It was published in Antimicrobial Agents and Chemotherapy this past October, and the senior author happens to be a disciple of actually Dr. Costerton. And his name, of course, is Paul Studley. And I'm always impressed with Dr. Studley's papers in that he has such incredible talent in imaging. And that's what we see in the lower portion of the slide. And what you see is Dr. Studley and his team are measuring activity of the culture. And so what we have here is a calcium sulfate impregnated capsule with tobramycin. Tobramycin is, of course, an aminoglycoside. And the way microbes resist themselves against aminoglycosides is they just send all hands to the pump and they begin to pump it out. And so what you see is as that antimicrobial will diffuse from the calcium sulfate bead in the center of that first image, we see a very high metabolic activity. And as time goes on and as the antibiotic diffuses, we see the action of the antimicrobial taking out the activity of the population. In other words, the patient is getting better. But by day 2, we see a nice zone of inhibition where there are no microbes growing. But by day 3, you can begin to see the emergence of these small colonies. And by day 5, we see the rise of the colonies. And this is a phoenix colony. And you're going to say, well, the antibiotic has just diluted itself below the MIC for Tobra. Well, that's not the case. And in fact, when you measure the tobramycin concentration in that zone of inhibition, it's still 5 times higher than the published MIC for Tobra for the particular organism that Studley groups characterize. So again, these phoenix agents are actually the microbes responding to the microbial in the microenvironment that they're present in. They're effectively doing an experiment in microbial ecology, reacting to what we and the host or the patient is actually doing to that community. So as you consider your antibiotic prescription regime, a continuous action will just breed the exposure or the resurrection, if you will, of these phoenix cells. So again, you see this is where we begin to change things up. We add an accessory electron acceptor. What Studley's group actually worked on was Pseudomonas aeruginosa, an organism that is resistant to many antimicrobials. And absent nitrate in an anaerobic environment, the microbe is again behaving similarly, but with nitrate, the antimicrobial was able to knock down the concentration of bacteria. So again, what this introduces is that oxygen tension can often influence the effectiveness of the antimicrobial and actually cause the resurrection of these phoenix cells. And it really is going to begin to confound our development of appropriate treatment regimes. So the bottom line is it's complicated. And in this algorithm that Studley and his team present to us, it really highlights and puts into perspective all of the things that we can think about. And when you fail to get culture data back from the lab, you can select resistant mutants. You can select for the presence of persister cells. VBNC cells may arise. And you can also have Dr. John, who we'll hear from in a few minutes, his favorite organism type, the small colony variant that is resident with the staphylococci. But now we have this new player added to the mix, and that these are the phoenix colonies. And the phoenix colonies, for all intents and purposes, behave as normal bacteria once the antibiotic is diluted away. The take-home message is that NGS could have told us who was there immediately. So I'm going to at least make the initial argument that coincident culture in NGS at the time you're beginning to work up the patient for an infection is really value added to your understanding and the development of an appropriate treatment regime. And so we hope you now appreciate, as we've gone through this very lightning round, the limitations inherent to traditional cultural microbiological techniques. And next up is how clinical specimens evaluated using next-gen sequencing can aid in your decisions in helping your patients attain a better outcome. So I think I'm going to stop there, and we're going to go to discussion.
Thank you very much, Mike. That was very, very illuminating.